Screencast intended to make up some of the time that we lost as a result of there being an ice day at the beginning of the semester. So this is going to take you through mitosis and meiosis, which is a relatively short little segment of material, but if we can get it taken care of online rather than in person, that'll save us a little time. The other thing is, this is a section of material that should be review for you for the most part. You should have gotten this in a high school biology course, or you should have been exposed to this in Biology 1124 perhaps. And I'm going to go through it in a way that really stresses the importance of knowing mitosis and meiosis from the perspective of how these two processes apply to genetics. So this is the beginning of a section on genetics. And so among the things that you should get out of this is knowing the difference between mitosis and meiosis, especially in reference to the production of gametes. Because ultimately we're going to be doing monohybrid and dihybrid crosses, and when we do monohybrid and dihybrid crosses, the first step of those crosses is to create the full suite of gametes that can come out of a given genotype, and that is done through the process of meiosis. So understanding meiosis is crucial to understanding genetics. And ultimately with genetics, we're going to be talking about dominant recessive alleles, different forms of inheritance, and how that affects genotypic and phenotypic ratios and allele frequencies. So this is all part of one unified part of this course that begins with transcription and translation, the central dogma, going all the way through genetics and how genetics influences evolution. So this is the middle part of it that bridges the gap between transcription and translation and inheritance style genetics. So this all fits into the general course. Once again, these are the 4 preconditions of evolution via natural selection, and these are the sections of the course that this applies to. And when we talk about inter-individual phenotypic variation being heritable, it is heritable because those genes are passed from one generation to the next in the context, inside gametes, and those gametes are formed through the process of meiosis. So you need to understand meiosis in order to get a handle on genetics. So let's go back and think about how genes are organized. And so genes, once again, are segments of DNA that code for a particular functional product. But this DNA is organized and packaged into chromosomes. So here in the lower left, we have a DNA molecule. And that DNA molecule is going to be wrapped around histone molecules. So the yellow portion here is a histone molecule. The purple is the DNA molecule shown schematically. Now, histone molecules are made up of amino acids that are largely positively charged. Remember that the sugar phosphate backbone of DNA is negatively charged because of the phosphate molecule there. And so this enables the DNA to stay associated with histone molecules. By wrapping the DNA around the histone molecules, those histone molecules can then be organized into thicker strands, and then these thicker strands are folded back onto one another and anchored in what ultimately becomes the structure of the chromosome. And so this makes for a very efficient way of storing DNA packaged by wrapping it around histones. One way of thinking about this is imagine a skein of yarn that you get at a craft store. You get it home, you take the wrapper off the skein, and slowly that thing will become unraveled. But one of the ways in which you can keep that ball of yarn organized is if you put it on a ball winder and wind it up into a ball that is more tightly, compactly stored, that then keeps that thing from coming unraveled. That would be a decent analogy for the structure of wrapping DNA around histones and then stacking these histones together to form a chromosome. So that's how DNA is moved about the cell and stored, is not just in raw DNA floating around in the nucleus, but actually DNA attached to histones organized into chromosomes. So in order to partition genes into new cells during cell division, the cell has to replicate its DNA. We've already talked about that when we talked about transcription and the replication of DNA. But at the same time that we do that, we also have to replicate the chromosome, because the chromosome is what's going to be carrying those DNA. So we do that during the S phase of the cell cycle, which we'll get to the cell cycle in a minute. But the terminology of chromosomes is sometimes confusing to students, so I just want to go through that first. When we talk about homologous chromosomes, we're talking about two chromosomes of the same type. So you have a blue chromosome and a red chromosome, but you see that they're both the same length. They have the kinetochore in the same place, the centromere is in the same place, and so along the length of the blue chromosome are a set of genes. Those same genes will be located in the same places on the red chromosome. The blue chromosome is perhaps inherited from your dad, and the red chromosome is inherited from your mom. When they're single sets of DNA bound up in a chromosome, we refer to them as chromosomes. But once again, after replication, the DNA is not only replicated, but also the chromosome itself is replicated. And so now we have a single chromosome, the blue chromosome, but this chromosome is now made up of two copies of DNA, and these two copies of DNA are on what we call sister chromatids. So there's a left sister chromatid here, and there's a right sister chromatid here. That's one chromosome of the homologous pair of chromosomes. Then on the red chromosome, we have a left sister chromatid here, and a right sister chromatid here, and that is one chromosome made up of two sister chromatids. So try and get straight the naming of chromosomes. This is a chromosome, this is a chromosome, this is a chromosome, and this is a chromosome. Now, this chromosome has twice as much DNA as this chromosome, but it's genetically identical to this chromosome. And this chromatid is genetically identical to its chromatid, and both of those chromatids are identical to this chromosome. So, chromosome terminology, it can be confusing. We can think about chromosomes as being a part of the karyotype, which is the entire chromosome complement of a species. This happens to be the karyotype of a human being. The numbering of chromosomes is done from the longest chromosome, chromosome 1, to the shortest chromosome, chromosome 22. The dotted lines are where the centromeres are. Centromeres are dense areas of what we call heterochromatin, chromatin that is really tightly packed such that it's very difficult for any gene expression to go on there, because the chromosomes are so tightly packed and the DNA is relatively inaccessible. And these are stained with a differential staining technique that allows you to see the banding. You'll notice here on chromosome 1 that you see these dark bands, and these dark bands appear at similar positions along the chromosome because these are the same kind of chromosome. Chromosome 1A, chromosome 1B. If these were your chromosomes, chromosome 1A might have come from your father, chromosome 1B might have come from your mother. On chromosome 2, we see once again similar banding patterns. That's because chromosome 1A may have came from your mother, and chromosome 1B, or chromosome 2A came from your mother, and chromosome 2B came from your father. So in humans, we have 23 pairs of chromosomes. One pair is what we refer to as the sex chromosomes, the X and Y chromosomes. The X chromosome is just like the Y chromosome. They have a centromere in the same place. It's just that the Y chromosome is missing big chunks of the genes that occur in the X chromosome. This has implications for the expression of traits that occur on the X chromosome, because in some cases, if there's a gene for a trait at the tippy end of this X chromosome, there's no homologue on the Y chromosome. And so even if that's a recessive allele, it's still going to get expressed because it doesn't have another companion there to block it or to mask it. This is what we refer to when we refer to as things being hemizygous. Hemizygous is half zygous. It means that there's a position for the gene on one chromosome, but that same position on the other chromosome doesn't exist, and so there's no complement to it in the homologous pair. Now, these are unreplicated chromosomes. When these chromosomes go through replication, chromosome 1A now has two copies, two chromatids. Chromosome 1B now has two chromatids. Chromosome 2A has two chromatids. Chromosome 2B has two chromatids. And so this is the unreplicated on the left and the replicated on the right, but it's the same genome, the same karyotype. We can describe these in terms of their schematic ways of looking at them. And once again, we can see the length is different from chromosomes, the position of the centromeres, which is where it's pinched in, are different, etc., etc. And if we move this Y chromosome up, we would see that this dark band corresponds to that dark band, and this dark band corresponds to that dark band. If I had made this figure, I would have probably moved those up so that the centromeres are aligned with one another. So that's the karyotype. Now, the cell cycle is a way of describing the life of a cell as it moves from cell division through a phase of growth to a phase of synthesizing the things necessary for cell division through a secondary portion of growth leading up to cell division, mitosis and cytokinesis. And so the cell cycle, the G1 phase, the S phase, and the G2 phase, these are all what we refer to as interphase. And then we have mitosis followed by cytokinesis, or the division of the cytoplasm. So there's initial growth phase, G1, a period in which the apparatus necessary for cell division is synthesized. That includes lots of things. It includes replicating the DNA, but it also includes replicating organelles that the new cells will need to function appropriately. And then the secondary period of growth, which is the final prep for cell division, is completed in G2. And then either mitosis or meiosis occurs at cell division, depending on whether you're doing somatic growth, producing new cells to make the organism bigger, or whether in meiosis you're creating gametes. And so we can contrast mitosis and meiosis in a table. The ploidy is referring to how many copies of a particular chromosome you have. So, for example, let's go back to the karyotype. In our case, we are diploid because we have 2 copies of chromosome 1. 2 copies of chromosome 2, 2 copies of chromosome 3. So when we talk about ploidy, N is the number of different unique chromosomes that you have, and the 2 refers to how many copies of those unique chromosomes you have. So in humans, we have 23 kinds of chromosomes, and we have 2 copies of that, giving us 46 chromosomes total. So the ploidy at the beginning of mitosis and meiosis is diploid. Every cell has 2 copies of all the chromosomes. The ploidy of the cells at the end in mitosis is diploid still, but in meiosis the amount of chromosomes has been reduced by half. It's only a haploid individual, haploid cells. The number of cell divisions in mitosis is 1. The number of cell divisions in meiosis is 2. Mitosis produces 2 daughter cells that are identical. Meiosis produces 4 daughter cells. They are unique genetically, and we'll discuss why that's the case. And the purpose of mitosis is somatic growth, and the purpose of meiosis is gamete production. So mitosis, this is a figure from your book, and this is just showing you the different stages of mitosis. And so in interphase, the nucleus is there, the chromatin is all strung out. You can't see the structure of chromosomes very well. But as you approach the onset of mitosis, during the first phase of mitosis prophase, you see that the chromatin, the material that makes up the chromosomes, this bonding of DNA to histone molecules forming chromosomes, these chromosomes go from being really stringy, like little thin threads, and they've condensed on one another. They've become thicker and thicker and thicker, and they actually become visible under a microscope. And this is the hallmark of prophase, is these chromosomes becoming visible. Now, when you look at the chromosomes in the schematic underneath, you see that you have 2 larger chromosomes and 2 smaller chromosomes. The 2 larger chromosomes are coded blue and red because that's 1A and 1B, and this would be 2A and 2B. And you see that they are not paired up with one another. They're just kind of floating around in the nucleus independent of one another. During metaphase, these chromosomes will line up along what's called the metaphase plate, and when they do that, they're not paired with one another. They're all still independent of one another. And you can see that in this micrograph up here. You can see that along the line here is where the centromere is, and the ends of the chromatids are hanging out into the cytoplasm. The structure of the nucleus, the membrane-bound nucleus, is broken down, and now you just have chromosomes floating in the middle of the cell in the metaphase plate, along the metaphase plate, and they're getting ready to be pulled apart. And the way they pull apart is they break right there at the centromere where the two chromatids are held together, and the two chromatids are pulled off to opposite ends of the cell. Once they're in opposite ends of the cell, the new nucleus begins to form, and you can see that this organism that had a diploid number of 4 chromosomes at the beginning of mitosis produces 2 diploid cells, each one with 4 chromosomes. So the ploidy hasn't changed. Diploid cells at the beginning, 2 diploid cells at the end. And then cytokinesis is just the separation of the cytoplasms after the nuclear material has been partitioned into these 2 new cells. So mitosis, a single episode of cell division, a single diploid cell giving rise to 2 new diploid cells that are genetically identical to one another because they're dividing the chromatids. This is once again how we grow cells. You started out as a single-celled zygote, and through the process of mitosis, you've grown into the trillion-celled organism that you are currently sitting at your chair or on your bed watching this screencast. So this is an onion root tip. This is a slide that you could see actually in lab. There's one of these slides or in your slide box in lab. And in this view, you can see cells in various stages of mitosis. You might want to take a minute and ask yourself, okay, what cell of mitosis are each of these things in? This is clearly prophase because you can see chromosomes becoming visible, but they're not organized in any particular way. This is metaphase because you can see those chromosomes lined up along the metaphase plate. These 3 cells are all in interphase. This cell is in interphase. This cell is in anaphase, where you're having the chromosomes being pulled apart into new centers that will ultimately become nuclei. This is telophase and cytokinesis, where you have 2 new nuclei and you have the beginning of a new cell wall. This is once again anaphase, the chromatids being pulled apart. Anaphase, chromatids being pulled apart. Anaphase, chromatids being pulled apart. So you could take any view and just go through the rest of this and say, okay, what phase are each of these cells in? That would be good practice in kind of cementing your ideas about mitosis. Meiosis is different in that this is how we make gametes, but it has the same 4 phases, prophase, metaphase, anaphase, and telophase, but you go through them twice. But the things that happen in prophase, metaphase, anaphase, and telophase are slightly different in meiosis. In Meiosis I, so we put 1s in at the end of prophase, metaphase, anaphase, and telophase for the first division of meiosis. In prophase I, you will see that the chromosomes have condensed, but you will also see that the chromosomes have come to be associated with one another. The red and the blue big chromosome are paired with one another, and the red and the blue little chromosome are paired with one another. That pairing is called synapsis, and that pairing is very important, and we'll get to the details of that in a minute. Those pairs, those homologous pairs, line up along the metaphase plate. So now rather than having 4 independent chromosomes along the metaphase plate, you have 2 homologous pairs lined up across the metaphase plate. And then in anaphase 1, it's not the chromatids that divide and go to new cells, it's actually the individuals of the homologous pairs. So the red chromosome from that homologous pair goes to one cell, the blue one goes to the other cell. And then you end up at the end of Meiosis I with 2 cells. Each cell has 2 chromosomes rather than the 4 chromosomes that you started with. So Meiosis I is what we refer to as a reduction division. It's where you go from a diploid cell at the beginning of Meiosis I to 2 haploid cells having half the number of chromosomes at the end of Meiosis I. Synapsis and crossing over is important because crossing over is where we generate new varieties of chromosomes. And so in this case, we have the two sister chromatids of the blue chromosome, we have the two sister chromatids of the red chromosome, and they're actually held together with this protein complex called the synaptonemal complex. And it basically sticks these two chromosomes together. Now, while they're stuck together, they can have the ends of the chromosomes break off and become reattached to the other chromosome, and this is what is referred to as crossing over. And so we have these chromosomes, they are connected to one another, and they tangle their ends up, and when their ends get tangled, they can break off and become reattached to the other chromosome, so that after crossing over, we began with a solid blue chromosome with two solid blue chromatids, a solid red chromosome with two solid red chromatids. Crossing over occurs so that in the mainly blue chromosome, we have a solid blue chromatid, and then we have a blue chromatid with two red ends. In the red chromosome, we have a solid red chromatid, and then we have a red chromatid with two blue ends. This is the result of crossing over. In the second phase of meiosis, so synapsis and crossing over happens in prophase 1, and the homologous chromosomes are partitioned to new cells in anaphase 1. And then when we get to meiosis II, we now have two cells going through division, but this cell division is very analogous to mitosis. We have the cells in prophase 2 with the chromosomes just hanging out with their two chromatids. Those chromosomes line up independent of one another along the anaphase plate. In anaphase 2, the chromatids separate, and those chromatids get partitioned into new cells, so that at the end of telophase 2, we still have haploid cells, two chromosomes, two chromosomes, two chromosomes, two chromosomes, but now there are four of them because you divided your haploid cell in prophase 2 into two new haploid cells. But you'll see that the nature of the chromosomes is different. This is a solid red small chromosome, mainly blue with red tip, solid red with blue tip, and mainly blue chromosome. Here we have red chromosome with blue tip, blue chromosome with red tip, solid red, and solid blue. So each of these chromosomal complements that are in each of these daughter cells at the end of meiosis, they're all genetically different because of crossing over. Now, when crossing over occurs, we refer to this as recombination. And the reason for this is, imagine that there's a gene right here on this chromosome. Imagine that that was the dominant allele for tongue rolling. Well, this blue portion over here would have also had the dominant allele for tongue rolling. Now imagine that down here somewhere we have a dominant allele for hitchhiker's thumb or something like that. Well, in this case, tongue rolling and hitchhiker's tongue or hitchhiker's thumb are both linked to one another because they're on the same chromosome. But because of recombination now, on this chromatid, tongue rolling and hitchhiker's thumb are still linked to one another, but now if the recessive allele for tongue rolling was on the red chromosome, this recessive tongue rolling allele is now associated with the dominant hitchhiker's thumb allele. And so these alleles, they're still the same number of alleles, but they're now combined in different combinations than they were before. This is why we refer to it as recombination. And so at the end of all of this, we have four daughter cells. Because of recombination, each daughter cell has a different suite of genes in it because of recombination. So, just to review, mitosis has the division of chromatids, which is exactly what is going on in meiosis II. So meiosis you can think of as a reduction division, reducing the total number of chromosomes that are in the cells, and meiosis II is just a mitotic division of those haploid cells. Once again, just single division, genetically identical cells at the end, two sets of divisions, genetically unique cells at the end. You can compare this to that table that we built back at the beginning of this section of material. So, mitosis and meiosis require duplication of genetic material and cell components prior to starting. Mitosis is a single division, producing genetically identical daughter cells, and only produces 2 of them. Meiosis is a 2-stage division that produces 4 genetically unique daughter cells. This uniqueness is due to both crossing over and the independent assortment of homologous chromosomes that occurs during anaphase 1. These genetically unique daughter cells from meiosis are how organisms create gametes. So that's a summary of this. It's that gamete formation that is important for understanding the initial steps of understanding monohybrid and dihybrid crosses, which is why I want you to know this. So here's a figure from a textbook, and there are some mistakes in this figure. It's a figure of meiosis. Take a few minutes as practice for practicing what you understand and don't understand about meiosis, and see if you can figure out what the mistakes are that are in this figure. This actually did occur in a textbook, and luckily it's a textbook that back in the day I did not adopt for use in a class because it had these kind of fundamental mistakes in it. So have a little bit of fun with this, figuring out what this artist did wrong. And that is it for this screencast on meiosis.